Okay, apparently I'm live now. So, hi everybody, welcome to my channel, and let's review some plant and animal form and function for Biology 304 for your test tomorrow. Now, before I get started, I did it again. Yeah, potatoes. It's probably just because I love to eat potatoes so much, and you dig them up from the ground. And as a kid, we used to grow potatoes, and we had to always dig them up, and they're look like they're attached to the roots, and they're stems. They're not roots. Even in my study guide, the syntax was right. Even though they're not, yeah, yeah, they're roots. I mean, sorry, I did it again. Potatoes are stems, and this is actually I'm going to address this. Uh, the reason why they're stems, it comes down to the structure of the vascular bundles in the vascular system. And this is a very important thing for the test because that structure lets you know whether something's a monocot, a dicot, a stem, or a root. And let me just show you really quick. I'm going to use the backside of my daughter's drawing of the family. I wonder who dad is, right? I got 3 girls in my family, and I wonder, dad right here. But let me show you really quick. Let's say we take the cross section of 2 stems, and you can see I got my cross section. And if I just quickly, if I have a U dicot and I draw like this along the edge, this would be the epidermis, this would be the vascular bundle, this is all the cortex parenchyma cells here. This would be a stem of a eudicot, which a potato is a eudicot. And then if I have it in the middle like this would be a root. Here's my epidermis. Here's my cortex right here. This would be the endodermis. And then right on the inside of the endodermis would be the pericycle. And then the larger cells forming a cross pattern in there would, of course, be the xylem. And then the smaller cells would be the phloem. That is how you tell the difference. So a potato is a rhizome, which is an underground stem. So I apologize for, it's because as a kid, I used to just dig them up all the time, and we called them roots. And it's like, you just can't sometimes get that out of your brain. Potatoes are, are right. Man, I knocked the whole thing. Are rhizomes, underground stems, just like an aspen tree. And in fact, that's a question on the test. If you see an aspen tree and they're suckering, and just right off the study guide there, if it's suckering, suckering, that's what we call just growing up from the ground. Those are from underground rhizomes. It is a form of asexual reproduction. So that's a test question. And the way you would know is you would dig them up, take a cross-sectional area, and look at the structure of the vascular bundles. If they're around, forming a circle around the edge near the epidermis, you got a eudicot, and it's a stem. The vascular bundle, whether it's a monocot or dicot, is in the center like that, you've got a root. That's how you know. That's it's as easy as that. I mean, of course, you got to make a pretty good cross-sectional cut so you can see it, but that's how it works. So now that I got that out of the way, I updated the study guide, the review questions, to make sure that, it was written correctly. And yeah, I apologize. It happens to the best of us. Now, you don't make that mistake on the test because I've clarified it now. But the one about the aspen tree, that's like word for word on the test. Okay. In fact, even drawing an illustration will be totally fine. So the way I want to run this review session is you can hit me up on the top chat. You can basically type in your questions. Top chat, I got some emailed questions. I'm going to cover those. You went ahead and emailed me. And the questions that were asked were pretty good. So one of them was if you could go over the structure of a eudicot and monocot. And one of the differences between them, there's several differences, and you'll need to know these. I'm drawing right now a monocot stem. And yeah, hopefully by the end of this semester, I'll be in my new office. It takes a long time when you build a building on your own. And I keep knocking it. I'll have a chalkboard or a whiteboard I can write on, so I don't have to keep doing this. But I hope to have my studio soon. Okay, let me write it on here. Dicot and monocot. Okay, if we were going to look at the stems, one of the differences between a monocot and a eudicot, there's multiple differences. One of them is right here. These little circles represent the vascular bundles scattered throughout the cortex, the pith here. Remember, the cortex, that is your parenchyma cells, right? These are live at maturity. They're storing things, they're making stuff. And if they're in a leaf, they're doing photosynthesis. This would be a eudicot stem. And you can see that the vascular bundles are on the outside. They're arranged in a circle. And there are differences in the roots too. I'm not going to give away every single answer, but that's looking at the structure of those vascular bundles is important for understanding the difference between monocots and dicots. Okay. And of course, you guys are on this. 2-3 emails about the potato. I covered it. I already said the one, number 11, you discovered aspen tree returns quickly from a forest fire. One person says they're growing from the roots. Another person says from underground stems. Remember, underground stems are rhizomes, and the way you tell a root from a rhizome is you look at the position of the vascular bundles. So if you're going to do really good on this question, it's 5 points. You explain the structure of the leaf. The structure of the stem. Explain the structure of a root. Point out the vascular bundles. Point out the different structures, and then explain here they're in this circle here. Okay. So diagrams, illustrations, you can do that for a test. It'll be really helpful. Okay. What's the difference between a primary and a secondary bud? The slides don't seem to use the language of primary and secondary bud, as in the same as apical and lateral bud. Yeah. So think about this. Primary is apical, right? It's at the apex. A lateral bud would be the secondary. It's one off of it. And this is a problem with sometimes, and I apologize for if I'm confusing on this. You go from one textbook to the next, or like the Wikipedia, and I love the Wikipedia. It can be an authority on this too. But I was using this high level book, and it referred to it a little bit differently than this textbook, right? So yeah, apologize for the difference in the terms, but basically the primary bud, that's where you're going to get a lot of your elongation of a branch is going to come from that. And then the lateral bud or the secondary buds, imagine you've got a branch and you got leaves coming off of it. I'm not very good at drawing here. Yes, this is my art project here. If I had a bud right here, this would be like an apical bud right there at the tip. This is where it's going to get primary growth, lengthening of the branch. Then right here, at the nodes where the petioles coming out the leaf, that's your lateral bud. And typically lateral buds, if they're close to the tip of the stem, don't form branches because this thing's actually sending out hormones down here to prevent growth. So if you whack that off, if you cut that off right there, those hormones stop, but then these lateral buds will actually grow into branches. That's the main difference there. Okay. I think that's it. So I'm starting to get some questions from you guys, and I'm just going to go write down these and see how we do with them. How does ATP synthase come into play between light reaction and the Calvin cycle? Micah, that's a great question. Think about, first of all, we got to put some big picture stuff here together. You've got photosynthesis, and the whole purpose of photosynthesis is to take the energy and light and some carbon dioxide, and we're going to store that energy and light as a carbohydrate by fixing carbon dioxide into an organic molecule. To do that, you need electrons, right? That's going to come from the splitting of water, and you need energy. You need both those things. So in the light reaction, this is all part of your test questions here, the light reaction occurs on the thylakoid membrane, right? So photosystem 2, this is where it starts, takes the incoming light energy, splits water, oxygen just forms, or The oxygen evaporates away, just floats away basically as O2. Then the electrons are elevated in energy. They go through the electron transport chain. Okay. As they go through the electron transport chain, phaeophytin, plastoquinone, cytochrome complex, that's a proton pump. And what it's going to do is it's going to take and start pumping protons. This for a thylakoid, it's going to start pumping protons into the thylakoid space. And then those electrons, they're going to get elevated in energy with photosystem. Now, here's this connection of ATP synthase and why that's important. ATP synthase is down here. Let's say we've got ATP synthase here. The protons are flowing through the ATP synthase, and this makes ATP. And as you remember, ATP is the energy currency of life. So during the Calvin cycle, as you are fixing carbon dioxide, the electrons are coming from the splitting of water. They get re-elevated here in photosystem 1. Then they go to, ferredoxin. And I always want to spell ferro. It's ferra, like F-E-R-R-E, ferredoxin. And then, NADPH reductase sticks it on NADP. And our electrons are carried to the Calvin cycle out here in the stroma, which if this was a chloroplast, this would be the stroma outside of this. And then this carries the electrons to fix it. So the ATP synthase, that's making ATP. So I'm taking the kinetic energy in sunlight, okay, and I'm transforming that to chemical energy by elevating those electrons. Those electrons go through the electron transport chain. And then when I create that electrochemical gradient, I transformed energy. I have energy potential right here. And then it goes through the ATP synthase, and that makes ATP. And the ATP is used in both the Calvin cycle. It's used in both the reduction phase and in the regeneration phase, where you make ribulose bisphosphate, RUBP. And that makes sense. It makes sense because if you think about ribulose, that's a 5-carbon sugar, like DNA and RNA. Actually, RNA, that's ribose. And then when you stick the phosphates on there, that's coming from ATP. So that's the connection. It's an aspen tree because of the way it is. You're right. Aspen trees are so awesome. The quaking aspens. Oh, I'm sorry, I'm shaking the whole thing. They're quaking aspen trees. They're so beautiful. Okay, which cycle is it connected to? I'm not certain here. The light reaction, the way I'm teaching it, that occurs in the chloroplast of most plants, and most of the time, the light reaction represents linear electron flow from water through photosystem 2, up in energy through the electron transport chain, back up to photosystem 1, to ferredoxin, to NADPH, to the 3-carbon compound, the C3, 3PGA, right, that you fix the carbon dioxide to. That's a linear flow of electrons because they're not going in circles. There is cyclic electron flow in some different types of bacteria, but the cycle is the Calvin cycle, and it's the flow of electrons that connect, the light reaction to that Calvin cycle, if that's what you're asking. ATP synthase makes ATP for the use in the Calvin cycle. Absolutely. So part of it is a light reaction. So yes, light reaction, that gets you you're getting from the light reaction your electrons by splitting of water, and you're elevating them in energy, right? So you reduce NADP to NADPH. This is your electron carrier. And then you take the energy in sunlight and you're transforming that to chemical energy via proton gradient. Then that's being used to make ATP, which ATP is used. That's the energy source for your Calvin cycle, because if you think about the overall reaction of photosynthesis, carbon dioxide plus water makes carbohydrate. The reactants, carbon dioxide and water, have way less chemical energy, way less potential energy than a carbohydrate. So you need a source of energy, and that's where the ATP is coming from. Could you explain 5 on the study guide about the trend and sizes of the gametophytes and the sporophytes? Yeah. So interestingly, this is something that's on the test is that alternation of generation. First question, actually, describe what makes a plant unique. And that's alternation of generations, which means that plants actually have two life forms. That in the primitive plants, we don't really like to use the word primitive, but in the earlier plants, plesiomorphic, right? You remember in ecology evolution, plesiomorphic old forms, the bryophytes, the gametophyte, which is haploid, is the dominant form, and the sporophyte, literally, if I drew a gametophyte, there's not really any leaves or roots here. If I drew a bryophyte like this would be the gametophyte, and then the sporophyte would grow right up on top of that. Okay, and basically, what happens over as plants evolved to live on land and become better adapted to living on dry land, what's happened is that the gametophyte has become smaller and smaller and smaller. So in the mosses, the bryophytes, the gametophyte, the haploid form, right? They're haploid, and they're making the gametes through mitosis. They're the dominant form, and the sporophyte is small. Then when you have the evolutionary jump forward to vascular plants, these are plants that have roots. They have leaves. They've got a vascular system to move materials between the roots and the leaves. At that point, in the ferns, also known as pteridophytes, the the gametophyte is only like this big. It's small. And the sporophyte, which is the diploid form that forms the spores through meiosis, uh, the sporophyte is much larger. But most of us here, in North America, New Mexico especially, when you walk out on campus or walk through the woods, and you see conifers like pine trees or spruce trees and Christmas trees. Not a real tree, but you get the idea. Or you see the cottonwoods or aspen trees, right? That's the sporophyte. Now the sporophyte in your seed trees of course make seeds and have pollen. Now the sporophyte is really large, and the gametophyte is tiny. And in fact, it's reduced to inside the pollen, the male gametophyte, and the female gametophyte is inside the ovules. So there are just a few cells. That's it. So they go through evolutionary time, they get smaller and smaller and smaller. Okay, can you touch on lateral meristem, secondary phloem, and xylem? If damaged, would the plant die? Well, definitely if the court cambium gets killed, right? Because if you take a tree and you girdle it, people do this to kill trees actually. If you girdle it, you cut away the bark and you cut into the court cambium. Remember, there's two types of this lateral meristem cambium, or this lateral meristem, the court cambium and the um oh gosh, the lateral meristem. What happens is that, or the vascular cambium, sorry. What happens is that the phloem can no longer transport material back and forth, right? So in that case, you cut the leaves off from the roots, and the tree dies. And if you cut far enough into the xylem, it will die because it can't transport water. However, one of the important things about the second, if you let me pull it up here so I can get it for you here. Whenever you have a tree, and a tree is woody, and they have a stump, they got the tree trunk, and that tree trunk is mostly secondary xylem, and it's called the secondary xylem because it's formed after the primary xylem, which is formed from the apical meristem. So the lateral meristem here forms both the court cambium and the vascular cambium. The vascular cambium produces uh, the secondary xylem. So if I've got a tree, we're gonna repurpose this picture here. You can imagine the bark is out here. This is the heartwood of the tree, and let's pretend for a second this is the vascular cambium. What's gonna happen is it's gonna lay down xylem cells. It's gonna grow xylem cells on the inside. That's a secondary xylem used for carrying water. And those cells usually die. They're dead at maturity, but they're just these tubes that transport water. If you take out the vascular cambium, that tree can no longer lay down secondary xylem. So while it may live for a while, the real problem with it is it can't grow any wider. You can't get secondary growth. And remember, primary growth is the tree growing tall. It's elongation. Secondary growth is growing wider. So if you damage the vascular cambium, no more secondary xylem, so that will severely limit its growth, if not kill it, but it can't grow wide. So if it can't grow wide, it can't keep growing tall because it would just topple over. Okay. All right. Can you touch on C3 and C4 pathways? Okay, so I don't know what you mean by touch on C3 and C4 pathways. As I need some more like specific questions here because I spent like 20 minutes talking about C3 and C4 pathways. So hey, Green, hit me back with just a little bit more of what do you want to know about C3 and C4 pathways? No problem. I don't have that in front of me. Type out your answer, man. I don't have 46 in front of me right now. I'm doing this mostly from memory here. All right. Can I explain why plants are green? There's a slide on that. And that slide says it's about the electron energies. Okay. The electrons can only exist at discrete energy levels, at discrete electron shells. And it comes down to, without giving away the answer completely, is that red light and blue light can elevate these electrons to discrete energy levels, whereas green light cannot. So that's a really, really solid start to begin that answer on of why it reflects green light. Okay. I'm a little bit confused about evolutionary trade-offs and angiosperms for having both the vessel elements in addition to tracheids. That's a really good question. It can be confusing sometimes why an angiosperm, a flowering plant, has both vessel elements and tracheids. Okay, so this is clearly part of the xylem, which is going to be moving water from the roots to the leaves. And gymnosperms, which are your conifers, they just have the tracheids. The tracheids are, I'm drawing them to the best I can, these long, narrow things with these perforations. And being narrow, they can transport lots of water if you have lots of them. But being narrow, one of the nice things is that they resist cavitation. The water column is less likely to break in them. So as an adaptation to a little bit drier or a warmer environment where there's less water, so these trees are less likely to get that cavitation in them when they become drought stressed. That's why if you look out in the Southwest up here, a lot of our trees are these conifers. Now, the angiosperms have the tracheids and the vessel elements. The vessel elements can be right next to them, but they're a little bit larger in diameter. And being a little bit larger in diameter, they can move more water. You can move more water and more nutrients. Guess what? You can grow faster. But there's a trade-off. If you're moving more water, you can grow faster. Great. But you are more subject to drought stress. You can, if you have more drought stress, then you can have cavitation and the tips of your branches can die. And in fact, if you walk around a UNM campus or anywhere in Albuquerque, one thing you'll notice is that a lot of the angiosperms, our elm trees, our sycamores, our cottonwoods, look at all the dead branches on the tips. That's from cavitation. A lot of water, those trees will grow very fast. But look at all the conifers. Very few conifers have dead branches. That's because they're more adapted. They're better adapted to the drought because they only have the tracheids, so they get less cavitation in there. So that's the trade-off. Okay, it's about, yeah, you can grow faster, but you don't do as well when it gets drier. Okay, I don't understand how to explain why a fern has a vascular system but no seed. Do all plants, seedless, go through all? Okay, Natalie, that's a really good question. All plants go through alternation of generations. All plants. That's a defining character of plants. First question. Make sure you include that in there. Okay, now a vascular plant. So as plants evolved, they were all these bryophytes. They lacked roots. They lacked leaves. They lacked the vascular system. The next evolutionary step was these ancestors to ferns. They evolved roots, leaves, and a vascular system, xylem and phloem. Gymnosperms, or the seed plants, which include gymnosperms and angiosperms, think flowering plants and mostly conifers, those seed plants evolved from other ancient vascular plants. So the evolution of a seed and pollen occurred about 310, 315 million years ago at the end of the Carboniferous when it kind of got dried out. So seed plants, which include both gymnosperms and angiosperms, they still have alternation of generations, but the gametophyte is much, much smaller. Okay, so it's like saying, vascular plants include gymnosperms, angiosperms, and those ancient ferns, the pteridophytes. It's also like saying, vertebrates include sharks, ray-finned fish, and tetrapods. And because we'll have a dorsal neurocord and a backbone, but yeah, our lineage came from lobed fin fish that broke off from the ray-finned fish, like 440 million years ago, right? So we're still related to ray-finned fish. We're all modified fish, but our lineage evolved on to become the tetrapods, just like the seed plants went on to go on and evolve into the gymnosperms and the angiosperms. Okay. And you're right. Ferns do reproduce through spores. Those spores will grow into a gametophyte, and then the gametophyte will then release gametes that will fuse, form the sporophyte. The sporophyte is diploid, will grow into your fern. Your fern, through meiosis, will then form the spores on the underside of those leaves. Those spores will then take off. They'll form the gametophyte, and it goes round and round and round. So, yeah. Okay, can you explain why does a root force water from the apoplastic route into cells at the endoderm? Why not just continue to the vascular tissue? Jackie, that's a fantastic question there. The plants would be able to control what is going into the vascular tissue, because once you get water and minerals into the vascular tissue, it's just going to go straight to the leaves. Sometimes plants, there's a couple of reasons here. One is that barrier can help protect them against letting heavy metals into the vascular system, which could damage the tree. They can block it. Rather, those metals might be flowing between the cells where it's not causing any damage, but if it gets into the xylem and goes to the leaves, it could damage the leaves. So it acts as a filter against some things that it doesn't want in there. It can also, keep out sodium. A lot of plant tissues and animal tissues as well try to keep out sodium. So if you've got a salty environment, keep the sodium out. It can also let potassium in. Just like our cells, we have a sodium potassium pump, pumping potassium in, sodium out. Plants also like to have potassium in them as well, because as we'll learn, it's really important for how plants can open and close their stomata, and of course do some other things that are important with potassium. And the other reason is in small little forbs. Forbs are these tiny little plants, like this. By forcing it into the root, forcing the water through from the apoplastic root into the endodermis, then into the the xylem, you can actually create some pressure and force water up the xylem, and in these more small forbs, you even get glutation from that. So that's one reason why. Okay, Elliot, C3 pathways, the 3 carbons and the 3 carbons out. Well, I'm not sure what you're asking, but a C3 pathway, what happens is that when you fix carbon dioxide, when Rubisco, which is the enzyme, fixes carbon dioxide to RUBP, RUBP has 5 carbons, carbon dioxide has 1. It fixes it, forms a 6 carbon compound for like a billionth of a second, and then it just splits into a 3 carbon compound, 3PGA. And because of that, that is now a 3 carbon compound. So you have your initial carbon fixation, which then goes on to form a 3 carbon compound, hence a C3 pathway. The 3 carbons out. Yeah, one of those carbons comes out. That's why we do you'll see Calvin cycle. You'll start with 3 RUBPs, 3 ribulose bisphosphates, and 3 carbon dioxides coming in at the same time. So you your 3 RUBPs. Each one gets carbon dioxide attached to it. And then for each turn of the cycle, with 3 coming in, you would get 1 3 carbon out. It actually takes like 3 turns to get 1 carbon out. That's why we balance it with 3 coming in, 3 going out. Okay. What's the advantage of PEP carboxylase initially fixing carbon dioxide in the mesophyll cells, and then you're using Rubisco to fix carbon in the bundle sheath? What's the cost? Man, that sounds like you just cut and pasted. All right. Okay, here it goes. It's all about photorespiration, right? So when plants become drought stressed, they close their stomata. And when you close your stomata, carbon dioxide is still getting fixed. Okay. Rubisco will still fix it. And as your stomata are closed, you're not getting gas exchange. No carbon dioxide's flowing out, no oxygen's flowing. Sorry, no carbon dioxide's flowing in, no oxygen's flowing out. So it's building up in there. You're getting more and more oxygen, less and less carbon dioxide. Well, what happens is at some level, when you draw it down, around 200, 150 parts per million, Rubisco starts fixing oxygen. Yeah, fixes oxygen. Actually costs plant energy. And that's called photorespiration. Well, so what the C4 pathway does is it uses PEP carboxylase to basically fix carbon dioxide in the mesophyll cells of a C4 plant. And PEP carboxylase basically has no affinity or very low affinity for oxygen. So the plant can close its stomata, keep doing carbon fixation, lower and lower levels of carbon dioxide. And then it moves that 4-carbon compound, oxalacetate and a malate, then goes into the bundle sheath cells where the carbon dioxide pops off. And then Rubisco can fix it through the normal Calvin cycle. So the reason why it does this is you take your initial carbon fixation and your Calvin cycle, separate them in space, so you can basically concentrate carbon dioxide in the bundle sheath cells so that Rubisco doesn't go into photorespiration, so you can close your stomata and keep doing photosynthesis in a drier environment. And of course, there's that's a benefit, and the cost is, well, think about PEP carboxylase, or think about PEP, phosphoenol pyruvate. We're not in biochemistry that really tells you a lot about what that molecule looks like, but importantly, it's pyruvate, 3 carbons with a phosphate group attached to it. That's an activated molecule. It takes energy. It takes energy to take pyruvate and stick an ATP onto it. So the cost is, yeah, you can keep doing it, but it costs you energy to recharge pyruvate to PEP, to phosphoenol pyruvate. So that's the main cost. Oh, already talked about the trade-offs of the angiosperms and vessel elements. That's on there already. Can you explain the difference between parenchyma, parenchyma, and sclerenchyma cells? I can, but we don't need to. Let's focus on parenchyma cells. They all form what is called part of the ground tissue. So in plants, you've got these 3 main tissues, right? You've got your dermal tissue. Think epidermis, endodermis. That's dermal tissue. They surround the plant, and the endodermis, of course, surrounds a vascular bundle inside the roots. You've got vascular tissue, and then you got vascular tissue. This is another complex tissue. Xylem, phloem, tracheids, vessel elements, sieve tube elements, right? All of these things. That's your vascular tissue. The third one is your ground tissue. The ground tissue is made up of those 3 types of cells: sclerenchyma, parenchyma, and collenchyma. Now, I don't have a question about sclerenchyma and collenchyma cells. I do have them in there about parenchyma, because the parenchyma cells form like your pith. The parenchyma cells form your the cells that do photosynthesis, the mesophyll cells, the spongy mesophyll, right? The palisade mesophyll cells. Those are in C3 plants. In a C4 plant, there are once again the mesophyll cells. That's where initial carbon fixation of the PEP carboxylase occurs. And then they also form the bundle sheath cells that surround the vascular tissue, where Rubisco is fixing carbon dioxide, doing the normal Calvin cycle. So the parenchyma cells, remember, they're part of the ground tissue, but they are where those are the mesophyll cells and the bundle sheath cells, and they're alive because they're doing photosynthesis, and they're alive because they're doing storage. So if you've got potato or you're storing pigments, they're having to move stuff in and out of those vacuoles. You're moving stuff in and out, and you're doing active transport. You need to be alive. So there you go. That's the parenchyma cells, and that's what you need to know about that. I'll kind of narrow that down a little bit for you. How did angiosperms lead to diversification of terrestrial animals? Well, yeah, I mean, they have flowers and fruits, right? And I've got a great series of slides, and that's just check out that series of slides on the first part of the class, and it's basically diversity gives rise to diversity. Flowers attract pollinators, right? So you have this evolution of all these different flowers and attracting all these different pollinators. You got pollinators coming to a flower. You got predators. You got predators waiting to nail all the pollinators. You've got seeds. You've got animals that are eating and dispersing those seeds and the fruits. So it's just diversity just gives rise to diversity. And in fact, a large majority of the diversity we have today is because of the radiation of angiosperms and flowering plants. The sporophytes also get smaller in time which attached to the top of the gametophyte, or do they get bigger? Oh, they're not attached to the gametophyte anymore. That's only in the bryophytes. I see, Melissa, where you're going with that. Yeah, the sporophytes are getting larger. Gametophytes are getting smaller. Think about like a flower. When you see the plant like a cactus and you see a flower on it or a rose, basically that plant, the rose bush or the cactus bush or the sycamore tree outside our classroom, that's a sporophyte. The gametophyte is so reduced that the male gametophyte is the pollen. It's inside the pollen grain, and the female gametophyte is like inside the flower. They're just a few cells. That's it. They're not growing on top of the gametophyte like they were in the mosses. Good question. Hopefully I cleared that up for you. Okay, Natalie, since phloem transports sugars back and forth, how is a xylem able to die at maturity if water transport is needed to create sugar? Very good question, actually. So here's a difference. In yesterday's lecture, I showed you that the movement of water from the roots to the leaves is a function of water continually going to places with lower and lower water potential. Water flows from high potential to low potential. So roots have a lower potential than the soil. The stem has a lower potential than the roots. The leaves have a lower potential than the stems, and the air has a lower potential than the leaves, right? Okay. The other thing also is so for this water to move through the xylem doesn't have to do anything. It's basically a straw. It's surrounded by cellulose, which is a hydrophilic molecule. It's a polymer. It's made up of glucose. So the water can stick to the sides of it and stick to itself. And so the water is adhering to the sides of it, and the water has cohesion, so it sticks to itself. Then when the water gets up to the leaves, it evaporates out of the leaf, and that transpirational pull helps pull water up the xylem. So there's no energy being done by the plant. There's no active transport of water through the xylem. However, let's see. So they can be dead. They're just creating a tube, right? Whereas the phloem, the reason why it has to be alive is because to move the sugars into the phloem, you got to move them. It's active transport. You've got all kinds of like primary, secondary active transport. You got symports, antiports, uniports all going on here, proton pumps. Those cells are doing this active movement of these things. And so to do that, you need energy to power those pumps. So phloem cells have to be alive. And we'll learn all about that on a Tuesday. I'm going to take a little sip here. Okay. What do plants do with light radiation? They do not absorb for photosynthesis. I mean, that's a really fantastic question. It just basically is transformed to heat, or it's transformed and just radiated back as heat, or it actually passes through the leaf and isn't used. The leaf doesn't use all of it. Man, that's a pretty good question. There's a lot of people that actually study like how that light radiation. Our sun is really intense at like 580 nanometers, but when it hits a plant, some of it's being reflected back as green light, and then other is heat, which is infrared. So it doesn't use it. Good question. Okay, a meristem. All right. Um, I'm gonna answer this, but another really good source for you guys, if you have a question like what is a meristem, man, the Wikipedia is just fantastic on that. But let me explain to you. A plant is it's a multicellular organism, just like an animal, just like you and me. And one of the defining characters of multicellular organisms is that we have differentiated cells that form different tissues, right? You've got skin cells, liver cells, muscle cells, nerve cells, on like 200 different cell types forming these different tissues and organs. Plants are the same way. And so we, plants and animals, all start out as a single-celled zygote. That's it. I mean, one cell. That's it. And then from there, it divides by mitosis. And then those cells, they get chemical signals and they start to differentiate into different cell types. Okay. So our stem cells in a human or an animal, they are undifferentiated cells that can, excuse me, that can grow into other things. They can differentiate into a stem cell can differentiate into a skin cell or a muscle cell or a nerve cell. It just has to start expressing certain sets of genes while turning others off. So a meristem is the equivalent of a plant stem cell. It can differentiate into like the procambium. If it differentiates into the procambium, then that can differentiate into the xylem and the phloem, the tracheas, the vessel elements, the companion cells, the sieve tubes, right? Or the sieve tubes. So that's what a meristem is. It's the plant equivalent of a stem cell. Okay. Why are gymnosperms going to be typically taller trees than angiosperms? That's a really good question. And some angiosperms tropics and can get up to 200 feet, but for some reason the gymnosperms are the largest trees in the world. And I actually don't know exactly why gymnosperms are able to outlive almost any angiosperm and grow so much bigger like the sequoia trees or a bristlecone pine. We do study, there are people that do study bristlecone pines, and they're really interested in the longevity of like, how can these trees live 4,500 years? And there's some really cool stuff that's happening at the cellular level where they can repair their cells so well, right? They have all these repair mechanisms. We have them too, but because of our terrible diets and not moving around very much, a lot of our repair mechanisms get tamped down. They get turned down. That's why like diet and exercise can kick those back up. But in terms for like how they're taller, I mean, I could come up with some reasons, but I'm honestly that's something that I'm not too good at answering. I'm gonna think about that because I've thought about that in the past, and I need to come up with an answer for that because that's a really good question. Do you need to know the exact compound names? No, don't worry about it. In the Calvin cycle, no. You could drive yourself mad trying to remember all these biochemical cycles. The thing about the Calvin cycle, I went through them and I did this in class on purpose. And my goal with that of showing you the compounds, what they look like, was to show you how the Calvin cycle worked through fixation. Like where's that carbon dioxide being fixed to it, and how it forms a carboxyl group, and then how NADPH actually adds electrons and hydrogens to reduce 3PGA, right, to G3P, and then how that how you take ATP and how you're going to regenerate your G3P back to RUBP. And there's a lot of steps I didn't even go into in that regeneration stage. But you do need to know like fixation, what's happening. Well, Rubisco, ribulose bisphosphate carboxylase oxidase, and it's a mouthful, but it tells you what it does. That enzyme, the most abundant enzyme on the planet, probably takes carbon dioxide, sticks it onto RUBP. That's fixation. Then you have reduction. That's where you add electrons to it. Because remember, carbon fixation, the Calvin cycle, that's a reducing reaction. We're reducing carbon dioxide, gaining electrons. So you have to have fixation. And it's an endergonic reaction, so it needs energy. If you think about the products, glucose has way more potential energy than carbon dioxide and water. You need a source of energy. That's the ATP. That's where you start to phosphorylate it, and you start by phosphorylating, you activate it, and it makes it easier to reduce and add hydrogens to it, and you get more. This is I didn't want to talk about this much, but you get more, non-polar covalent bonds, which store more energy than the polar covalent bonds. So you need to focus on fixation, reduction, regeneration. That's the important part. All right. And then know why there's 3 coming in and 3 coming out. And that's it. Don't worry about trying to memorize all those names. Will there be a question about CAM on the test? No, actually there isn't. I actually didn't get a CAM question on the test. I don't even know if I covered it much more than like 2 minutes on class. But there is that question about the advantage of a C4 pathway. Can you explain why vascular bundle is located in the middle of a root? That's a really good question. And the idea that one of the reasons why it's in the middle of the root is so that the plant can kind of control what gets into the vascular system. Root pressure is generated. Root pressure is generated as water flows into it. You have to have high water potential in the soil, right? And as water flows through the root, through the symplastic or the apoplastic route, it goes through the endodermis, Casparian strip, and it will continually flow in there. And because the water potential is really high, think like it's a hypotonic solution. Water keeps flowing in there. And if you remember the cells will expand, right? Well, the root's doing the same thing. So the water goes in there, but those cells they're surrounded by these rigid cell walls that don't really expand that much. So the water gets forced up the plant, right? And if it's enough water pressure, you can have guttation. You can force it right out of a closed stomata. I have a question about this question. What are the trade-offs of angiosperms, vascular systems, and gymnosperms? Alexander, I actually covered that in detail a little bit earlier, but basically it comes down to how the gymnosperms have better adaptations for drought stress or lower water environments, and angiosperms can move more water so they can grow more quickly, but they face cavitation, and I talked more about that earlier. Why is the vascular bundle inside the epidermis in the middle of a root and not near the epidermis like a stem? I want to say this is related to photosynthesis. Well, like I said, a lot of it has to do with, the root controlling and able to control what gets into the vascular tissue. As I said earlier, it's important. A lot they're like metals and soils, and metals, if they're just going through an apoplastic route, might not have a big effect on the plant, but they get into the vascular system and they go all the way up into the leaves, then those metals might affect photosynthesis. They might affect key metabolic processes and can affect them. So part of it is having the endodermis around that vascular bundle in the root allows that root to have a little bit more control of what gets into the rest of the tree. All right. Man, it's been almost an hour, 48 minutes here. So any other questions? Are we all good? Was this helpful? I hope so. All right. Well, I see about 59 of you still on. How do farmers deal with salt accumulation in their fields? Oh, there's Willow saying hi to me, and then of course my little buddy Titus. Oh well, yeah. Yeah, whatever. Man, that potato thing, we're done with that. I know. Yeah. Hi, Willow. What am I doing? Yeah. Well, at any rate, yeah, you guys are welcome here. All right. Yeah, kid, wife, dog, they're all coming in to say hi. She wants to see what daddy's doing. Yeah. Oh, she wanted to be in here. At any rate, okay. Yeah. We'll see you later. Good luck on the test tomorrow. I hope this was helpful for you guys. I'll post a link in the in Canvas. Whew, tired here. This is my second review session tonight. I had another class. Yeah, you guys are all welcome. Yeah. I don't know. It's like 8 pages long. Yeah, it's a 23 questions or multiple choice. There's matching. I went over that in class, actually. I went over that. So yeah, you guys are all welcome. Yeah, you definitely. And thanks for joining in. You know, I'm glad that so many of you, you know, over a third of the class, almost a half the class showed up. So yeah, I will continue to do these reviews like this for the next 3 tests. And you know, the first test with a new professor is always hard. It really is, because you don't know like what the test questions are going to look like, you know, what the level of expectation is. Yeah, I gave you that study guide, the review questions, and I mean, I literally made the test off of it, sent it to the TAs. Your TA Emily gave me a lot of feedback, and she said, you know, like, yep, this test is what you've definitely taught. So that's good. Okay, got one more question. I don't know any short answers. There's like 10 short answers on there, something like that. I don't have the test in front of me. Okay, but in the act of growing root, which zone is the zone of cellular differentiation? Oh, that's just if you got the root tip, it's behind the root tip. Just go look at that slide. You can see where you got the differentiation is the zone of maturation. That's where the cells are actually maturing, and that's where cell differentiation occurs. So if you have, for example, a procambium cell, that's a type of meristem, it will start to differentiate into like the xylem and the phloem. It'll differentiate into like sieve tubes and companion cells, and then it will differentiate into like tracheids or vessel elements. All right. You guys are good. All right. I'll see you in class tomorrow.